Hey, welcome back to our YouTube channel, the number one podcast in the world. Thank you so much for listening and subscribing. I'm Kavindya, your host, and I'm joined by my other group members. I will introduce the members who will participate in the podcast. First, let me introduce Aravinda, then Akshan Balasuriya, next Binadi and then sonara. This podcast continued by about name co-host. Today we are going to talk a podcast about genomics. After listening to the podcast, you will be able to know many important things about genomics. What is the genomics? Fun facts, example of genomics, danger of CRISPR, final conclusion about genomics, like that. So don't forget to stay with us until the end of the video. If you like our podcast, subscribe our channel, like this video and comment below. Okay, I would like to pass the opportunity to Binadi to present an introduction to genomics.
Thank you, Kavindya, for giving me this chance to speak. Now, I'm going to introduce you our topic. As Kavindya said, our topic is genomics. Note this also, I'm saying these things as simplest way I can. So, it will help to understand this topic to beginners as well. If we want to learn about genomics, first we should identify what is a genome. A genome is a set of instructions needed to create an organism. They are stored in our cells, same as DNA. DNA is an amazing molecule that stores information using a special code. You can think of a genome as an instruction list to build and grow an organism. For instance, you who is listening to this podcast right now. The human genome contains about 23,000 instructions that coded for different proteins. Each of these instruction or functional unit of DNA is called gene. The study of how DNA works is called genetics. So now we are slowly coming closer to our main point. It's genomics. From the things we discussed earlier, we discovered what are genomes and the environment of genomes. So, the study of the genome and its environment is known as genomics. I'll say it again. The study of genome and its environment is known as genomics. Now I think you all have an idea about our topic. Listen till the end if you want to be an expert in genomics. So, if I turn to our topic again, I know everyone is curious about genomics history. Trust me, it is way longer than you think. So, let's look at the history of genomics. The first step was in 1953. Mr. James Watson and Francis Crick discovered of DNA structure which laid the foundation for understanding the genetic codes. After that, Human Genome Project. We can say HGP as well. So, the HGP was an international research effort launched in 1990 with the goal of squeezing the entire human genome. It was completed in 2003, providing a map of human genetic information. Then, after 2 decades have passed, some of experts discovered functional genomics. Beyond squeezing, functional genomics aims to understand the functions and interaction of genes and their products. Techniques like transcriptomics, proteomics, and epitomics help unravel gene expression pattern and regulatory mechanism. As a recent update in genomics field, we can say genomic editing. The development of CRISPR-Cas9 gene editing technology has revolutionized the field, allowing precise modifications of specific genes. This has immense potential for both research and therapeutic applications. So, this is the history and present situation of genomics. I know Now you all are craving for know more details about genomics and CRISPR. Yeah, genomic topic is that much interesting. So, I will pass this chance to Miss Sonara. Furthermore, she will aware you about genomics and CRISPR.
Yes, thank you, Binadi. Now, I'm here to share some CRISP trivia with you. Did you know that CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats? CRISPR is a long term that was found by Japanese scientists studying E. coli bacteria and their immune system. It is also a natural defense mechanism in bacteria and archaea that allows them to recognize and eliminate invading viruses or other foreign genetic material. Also, did you know that CRISPR can alter DNA, including that of human embryos? However, using CRISPR to edit embryos that become newborns is normally not approved due to ethical and safety issues. However, many countries have laws against this practice. It is also experimental and cannot produce healthy infants. The scientists who discovered and developed CRISPR as a gene editing technique were awarded the Nobel Prize in Chemistry in 2020. CRISPR-Cas9 is the most prevalent type of CRISPR, and it uses the Cas9 protein to cut DNA at specified sites, allowing for gene editing. CRISPR technology offers the ability to heal genetic illnesses, develop new crop kinds, and perhaps abolish some diseases. The ethical ramifications of CRISPR are still being debated, particularly with regard to the possibility of human genetic editing. However, one potential scary fact about CRISPR is that it has the potential to be misused for unethical purposes, such as creating new bioweapons or genetically modifying humans for non-medical reasons. Additionally, the potential long-term effects of altering genes through CRISPR are not fully understood, and unintended consequences could arise, such as unintended mutations or unintended genetics and social impacts. It is essential to continue a regulated approach to the research and use of CRISPR technology to prevent such risks. Thank you. Now I will give my turn to Aravinda to talk about his work.
Thanks, Kavindya. So, talking about CRISPR, as you can see, the nature of this technology actually raises the question how ethical this technology actually is, or is genomics as a field to be studied going in the right path towards advancing humanity, or would it end up taking humanity away from humanity itself? So let's dive deeper into this idea and let's talk about the philosophical reality, the dangers of CRISPR, and etc. So, talking about ethics, I actually saw this news article about CRISPR being used in China to get rid of HIV/AIDS in babies, and it actually ended up producing babies not only without HIV but higher than a normal IQ. You can actually look it up on Google or whatever. And this is really fascinating and scary at the same time, because like imagine if humans got the full control over humanity, it would, in my opinion, mark the end of humanity itself. As like what defines humanity itself is the lack of control we have on ourselves. It is what that makes us human. So imagine if we got full control over the nature of our offsprings, if we could decide their height, if we could decide their skin color, if we could decide everything about them, wouldn't that be unnatural, and wouldn't that kind of rip apart the social fabric itself? And this in my opinion raises the biggest ethical concern, because it used to be, CRISPR actually used to be a tool that people used to like experiment with to get rid of lactose intolerance or like peanut allergies. And now people are kind of trying to use it to create like superhuman babies. So what are the limits of CRISPR? And I feel like this kind of technology could be even used to create like create resources for wars in the future. Like for an example, this kind of technology can be used to create super soldiers in a way. I know it sounds really weird, but like it is a possibility. And this is the biggest ethical concern with CRISPR, and what kind of a thing CRISPR is actually capable of doing. Is it a simple tool that's going to kill lactose intolerant, or would it be used as a tool to like create super soldiers in the future? We don't actually know. And yeah, Akshan.
Okay, now we have reached the end of this podcast, and let me address you the main question you might have encountered while listening to us. Why, on the podcast about geometrics, did we choose CRISPR and its effects? The reasoning is pretty straightforward. CRISPR is one of the most important, innovative, and the most groundbreaking piece of technology in this field of geometrics. It literally overshadows everything else in the field. We are pretty sure that CRISPR would precisely change the world, and that's the main reason why we choose to focus so much about this tech on the podcast. And you might ask, what is the conclusion we can reach from this information? While we explore the world of geometrics, we also opened our eyes to the reality of ethical problems that might come with the upcoming technologies in the stream that might supercharge or even kill the future of medicine and humanity. While we do celebrate the worthy attention progression of CRISPR, we must also stay observant in addressing the ethical implications. And there we go. We have reached the end of this amazing journey of geometrics and its future. We hope you had a nice experience listening to us. Adios and have a good day.